Hello everyone. In this video we are going to study about different types of epithelial tissues present in human body. So we know that there's a different type of epithelial cell layers or epithelial tissues. They consist of are made up of type of sheet of some aggregated cells like you're seeing in this different images of different type of epithelial cell layers, and they are covering the different surfaces like outer surfaces of our body, and some types of epithelial tissue layers are present in our internal hollow organ, viscera and other structures. So they are arranged outer side cell layers, body surfaces, as well as inner side. According to simple epithelial tissue, they are in a single cell layer having single epithelial cell layers resting on a membrane which is called basement membrane. So this is under simple epithelial tissue, and this is the simple cuboidal epithelial cell layer. And same wise, under the simple epithelial tissue, this is the second example of the simple columnar epithelial cells. They are also single cell layer, but the shape of cell, these are the elongated, vertically placed. The vertical height it is more, but in cuboidal it is almost square in shape. So this is the simple epithelial tissue. Next example of type of epithelium, it is the stratified epithelium, and again they are resting on a single basement membrane structure. They are single cell layer, but cells are very flattened from top to down, and at the end they are very tapered. So this is the example of simple squamous epithelial cell layer. And the second example, that is the stratified squamous epithelial tissue layer. Is in this diagram, the multiple cell layer, more than two, they are present, and this is under the stratified epithelial tissue example. Next, in this diagram, this is the example of pseudo-stratified columnar epithelium. Why it is called pseudo? Pseudo means false. False. What false? That we are watching in this diagram, that the position of nucleus is not aligned, but they are placed on uneven levels, and this position of nucleus in uneven levels, they looks like a multiple cell layer. So this multiple cell layer appearance, false appearance, it is called, that is why pseudo stratified columnar epithelial, and it is composed of one layer of the cell but looks to be multi-layer due to the different cellular height and different nuclear positions. And in case of transitional epithelial tissue layer, the cell layer, it is composed of multi-layer of cells, and it is characterized by surface most layer, means the topmost layer, it is almost dome like shells, means their upper top surface, it is forming the dome shape or arced pattern like structure, and when we see from the top side, it looks like a umbrella in shape. And the transition is occurs on the basis of shape of cells from basement, then to mid, then to top. And we can see starting from the basement cell layer, the shape of these cells are very different, almost square or same shape, vertical and transverse ratio. In the middle region, the shape of this cell, it is little bit different, and on the top it is dome shape. So in this multi-layer transitional shell epithelium, neither they are looks like a squamous or neither they looks like a columnar. So cell shape, they are changes from basement to top, and this is the transition of features of cell appearance from basement to top, and this is the transitional epithelium. Remember, in the case of transitional epithelium, the topmost layer will be dome like cells, umbrella in shape especially. First step during the microscopic study of different epithelial tissue cell layer, you might know what structures you should identify from each epithelial tissue. The three main things you should follow during the observation of microscopic study of the different epithelial tissue. First point, you should first emphasize on the layers of cells of the sample tissue. Means you need to check that is it a single cell layer or double cell layer or multi cell layer, or which type of uh, tissue it is, or which type of epithelial cell it is. So cell layer identification, it is must during the microscopic study of any epithelial tissue. Second point, the different shapes of cell provided under the sample. We should check the the epithelial cell, it is cuboidal in shape, or it is somewhat flattened in shape, or it is vertical in shape on the lower side, or it is present in uneven shape or uneven locality of the nuclei, or in case of transitional epithelium, the different shapes of cell present from the basement to top. So second point, we need to focus on the different shapes of cell of the provided sample. And the third and main thing, we should check the basement membrane and the lamina propria of any epithelial tissue. We should always observe where the basement membrane is present, and just below the basement membrane the lamina propria region will be present, and we will see in the next image. So these are the main three things: layers of cells, shape of cells, basement membrane and lamina propria. We should check during the histological or microscopy observation of different epithelial tissue layer. So first in this diagram, you can observe the simple squamous epithelium identification points and how the simple squamous epithelium looks like. So see in this diagram the circular pattern. These are the part of the alveoli. These are the magnified image of the lining of lung alveoli, lung tissue. So in this lining of alveoli, when you magnify, and you can see the presence of cell, they are very flattened and they are single cell lining. On every border of the alveoli, and this is the simple squamous epithelial tissue. How you can identify that this is the simple squamous epithelial tissue? It show a single cell layer rest on the single basement membrane. Second point, the cells of this simple squamous epithelial tissue, it is very flattened. They are very flattened, just like a plate, and having considerable length, means it is somewhat extended in length from side to side, but they are not much in height. So flattened cells are present basically. And third point is the nuclei in this simple squamous epithelial, almost they are present in the center, and somewhat flattened nucleus is present in the sample tissue, because they are simple squamous flattened type of cells, so nuclei it is also somewhat flattened in shape. Hence we can identify the simple squamous epithelium, how the cells are flattened, how they form the single cell lining, they present above this single basement membrane, and the nuclei is also flattened. So this type of epithelium, it is present in lung. Or the other side, to check this lining, this type of cells, like the pleura of the lung, like the internal lining of your abdominal cavity, that is the peritoneal cavity, the lining of the blood vessels also, they are formed with the simple squamous epithelium. So this is the diagram of thyroid follicle, a single follicular body showing the single lining of cuboidal epithelial cells, and other side where we can identify, we can found this simple cuboidal epithelial cells like kidney tubules, like in proximal convoluted tubule, distal convoluted tubule, or main collecting tubule, like thyroid follicle. This is the diagram, the germinal layer of the ovary, outer lining of the ovary, and some pigmented layer of the retina. These are the sites where we can check the cuboidal epithelial tissue, simple cuboidal epithelial tissue. In the next diagram, we are watching the simple columnar epithelial cell layer, and we can see the each cell, it is vertically in more, having more height, and transverse is very less, and they are arranged parallelly from one by one. So this is the appearance having simple columnar epithelium, and they have the two main identification, that maybe they are present in the ciliated form, so they will be called simple ciliated columnar epithelium, like we can tract in a respiratory tract, uterine tract, uterine tube. And the second is non-ciliated columnar epithelium, and this type of we can trace in a stomach, in a gallbladder, in intestine. This is the diagram of simple non-ciliated columnar epithelium, and having no cilia on the top, and their position of nucleus, it is towards the basement membrane. This is the basement membrane, and nucleus is present below the lowermost area, means lower 2/3 area, and the top 1/3 area, it is having full with cytoplasm. So vertical arrangements, and they are compactly arranged, align themselves, they are simple columnar epithelium. So this is the diagram of simple columnar ciliated columnar epithelium, and we can see the cilia. Cilia are just like a fine thread like cytoplasmic procession, and present on the each cell layer on their top. So these are the simple ciliated columnar epithelium, and this type of cell layer present especially just like a respiratory tract, like uterine tube, fallopian tube mainly. In the next image, we are checking the non-keratinized stratified squamous epithelium. In this case, the squamous cells are present in multi-layer form, and the nucleus is present in the whole row of the cells from the basement till the superficial most layer. The difference is only the nuclei present near the basement membrane. They are almost spherical and rounded, and when you reach on the topmost layer, the nuclei becomes somewhat flattened. So non-keratinized type of epithelium, they are present inner lining of the oral cavity, the inner lining of the vaginal cavity, the outer surface of the cornea, and the lining of the esophagus. These are some site just like anorectal canal area, and all these are the site where we can found the uh, non-keratinized stratified squamous epithelium. The purpose of this type of epithelium is to avoid the so they can bear the friction type of movements. In the next diagram, the keratinized stratified squamous epithelium means the topmost layer of this type of epithelium, they have almost dead cell layers, means they have new nuclei present in their cells on the topmost area, and the topmost layer they are most flattened, and near the basement membrane area the nuclei are almost spherical. When we reach from the basement membrane towards the superficial level, the nuclei become flattened and then they disappear on the topmost multiple stratified layers. So this keratinized stratified squamous epithelium we can observe on the outer surface of our skin, like thick skin, thin skin, etc. Next image, this is showing the pseudo-stratified columnar epithelium, and in this diagram here the basement membrane and just above the single cell layer lining, but the nuclear position they are different. Some nuclei present near the basement membrane, some are in the middle position, some are present on the topmost. So nuclei level are they are not aligned, but this cell is single cell layer, and due to this false appearance, false positions of the nuclei, due to this they looks like a multiple in cell layer, but they are not. So this is called pseudo stratified epithelium, and pseudo word means the false, and this type of the cell they can be ciliated or non-ciliated. In this diagram this is the non-ciliated pseudo stratified columnar epithelium. Next diagram showing the ciliated pseudo stratified columnar epithelium means the nuclei level are present in different level, but they are in a single cell layer form. On the top of each cell there fine cilia, the cytoplasmic projection are present, and this type of pseudo stratified ciliated columnar epithelium we can trace in a inner lining of the respiratory tract and like a tracheal area. And next in this diagram, this is the diagram of transitional epithelium. Transitional means the shape of this cell are changes from basement, then towards the mid, and then to the top, and the topmost cells almost they are dome in shape, umbrella in shape. So transition occur on the basis of shapes of cell from basement to top layer, superficial layer. That is why it's called transitional epithelium, and this is multicellular layer. This is not in single cell layer. And this type of epithelium we can trace like a inner lining of urinary bladder, the ureter,